In this video, we're going to talk about some trial preparation considerations. You might have noticed that the basic trial events, like the presentation of the images to the participant, are not directly inside the trial sequence. Instead, they are placed inside of a sequence called recording. This is because we need to do a little bit of trial preparation before we actually start recording eye movement data and execute the important trial events. So, on each iteration of the trial sequence, we first do a few things to get ready for the trial. First, we use a prepare sequence action to load any experimental stimuli that will be presented to the participant into the display PC's memory buffers. This way, when it is time to actually present the stimuli, the computer already has the files associated with these stimuli loaded in its memory. In this project, the stimuli only consist of image files, but the prepare sequence action will also pre-buffer video and audio files if a project uses them. The prepare sequence action also allows us to transfer an image from the trial to the host PC so that the experimenter can have online feedback regarding where on the image the participant is looking during the trial. We'll discuss this feature further in a later video. Next, the drift correct action will present a single fixation target on the display PC monitor. The position of that target is determined by the X location and Y location properties of the drift correct action. In this case, it will be presented in the center of the screen. During the experimental session, the experimenter will see a representation of the display PC monitor, including the position of the fixation target on the host PC monitor. When the participant's gaze is steady on the fixation target, either the experimenter or the participant may press the spacebar on the keyboard to start the trial. Note, the drift correct action is optional, but often it's a good idea to include it in your project. If the experimenter wants to recalibrate the system, then instead of pressing the spacebar, the escape key can be pressed to bring the two computers back into the camera setup mode. From the camera setup mode, you can then recalibrate the system. Once recalibration is complete, the project will pick up where you left off. Once the drift correct action ends with the spacebar press, the project will enter the recording sequence. The recording sequence of the project should always have an iteration count of 1. This means that the recording sequence is not used to do any looping. Remember, the trial sequence handles the looping through the various trials of the experiment. So, even though the recording sequence is a sequence node like the block and trial sequences, it's not used to do any looping. Instead, it is only used to send a command to tell the eye tracker to start recording eye data when the experimental flow enters the sequence, and to send another command to tell the eye tracker to stop recording eye data when the experimental flow exits the sequence at the end of the trial. It sends these commands because its record property is checked. So, whenever you have a sequence whose record property is checked, the host PC will start recording eye data when that sequence is entered and stop recording eye data when that sequence is exited. Please note, you almost always want the recording sequence to be the most nested or innermost sequence of the project. For the recording sequence, we also check the property is real-time. This elevates Experiment Builder's priority as an application on the display PC so that other processes that might be occurring on the display PC cannot interfere with the Experiment Builder project. Timing is critical when we are performing the actual trial events, so we want to make sure to optimize the performance of the display PC for this part of the project. If the basic setup for preparing a trial seems to involve a lot of things that might be difficult to remember, please remember to check out the Experiment Builder project checklist that is part of the user manual. You can access the checklist by going to Help, Contents. This checklist serves as a reminder for how to do things like trial preparation, and it can help you to avoid headaches in data collection and data analysis.